computer and DNA are quite similar. And also I'm doing automation, so I'm kind of in the bridge. I'm bridging the computation, I'm bridging the biology. So automation kind of pull these two fields together. In my perspective, yeah.
So the DAMP lab, the Design, Automation, Manufacturing, and Processes lab, is a core facility on the Charles River campus that provides microbiology services to people. So I founded the DAMP lab because I needed the DAMP lab to exist. If I want to make software tools, and I get you a software tool, and you design a DNA sequence or a novel organism, it has to get off your computer. It can't be one computer, one lab. It needs to be many computers to one lab. So what we need are people who have that shared vision of a model where the software and hardware can interact, where we can make software modifications and the hardware can be responsive to that and vice versa, and have a platform. And really, Opentrons has always been that kind of partner.
When I was in college, my biology teacher kind of mentioned to me the first time the Human Genome Project. I knew I kind of want to do something with engineering, and I run into DAMP lab. And it's the first time I really heard the word synthetic biology. The first project I did was for genetic circuit. Basically, you modify the bacteria genome so it will react like a biological circuit inside the bacteria. I changed the functionality of this organism, so it's very cool to me. People come to us, they tell us what they want. In the end, they don't really have any automation experience. The cardiomyocyte project, the research goal of that center is to use the amount of heart muscle tissue we produced to build a fully functional heart. They have 3D printed smaller artifacts, so the muscle tissue will grow around it and it will contract. Like a heart pumping, it will actually contract. Mice hearts are this big, and human hearts are significantly larger. So instead of human actively sitting in front of the tissue culture hut, robot take care of that work. It take her hours and days to do it, the same amount of time the robot do it. It can increase the throughput more than 10 times. Don't look at the stuff on the top because I'm still trying to figure out what they want.
I'm gonna judge you so hard, Rita.
Yeah, I do highlights, I cross things out.
The pilot projects that we did, the goal was to try to hit as many different departments as possible. So we had some in BME, we had some in biology, physics, bioinformatics.
The microbiome project, basically what we're doing is next generation sequencing is more applied to detect if there's any pollution inside the ocean. There's also a regular soil sample to see if there's any regular pollution in the forest. And this completes the calibration. Now we're about to start run. So Flex is definitely easier to use. It has a lot more space. It has a lot more deck space for different hardware module, additional labware onto the deck. Oh, so now it's my time to relax.
The founders and folks that were doing this were tinkerers. I was kind of envious of the fact they had the ability to tinker around and get these things to work. So we started with the OT1, we had an OT2, now we have the Flex. So with the newer models of the robotics that we've seen, the precision and accuracy of those robots has gotten better. The ability to program those robots has gotten to be more flexible and more robust. And then also the engagement with Opentrons has become more regular and meaningful. If you look in the 1960s, how many people touched a computer? Relatively few. If you look now, you've got a computer in your pocket. So the idea would be, hopefully in the future, people will interact with biology as much as we interact with computers. It's we're not going to have a test tube in our pocket, But we could have a program that's designing something biologically or innovating something biologically, and we're interfacing with that. Giving more people access to biological manufacturing will democratize biological design.